There are a number of situations where it's possible for there to be chromosome abnormalities in an individual. And these chromosome abnormalities can fall into a number of different groups. But one of them is that the individual ends up with the wrong chromosome number. So recall that every species has kind of a set chromosome number, and for humans, that should be 46. But it is possible that sometimes you do end up with a human that has something other than 46. So they might have 45 or 47 or 48. So some kind of slight variation from that. Well, the way that this occurs is that this is based on an abnormality or abnormal meiosis. And specifically what we're talking about causing this is that the chromosomes do not separate as they should. And this would be during either anaphase 1 or it could be during anaphase 2. So both anaphases, or really any anaphase that we have, is supposed to separate chromosomes from each other. So if the chromosomes fail to pull apart from each other, you end up with an inconsistency between the number of chromosomes that you have on the two sides of the cell. So we do have a name for this inability or this inconsistency to pull apart. This is called non-disjunction. So non-disjunction would just be this failure to separate. And again, the end result is going to be that we end up with a cell that has too many chromosomes inside of it. So if we're talking about during anaphase 1, just to draw that, recall that during anaphase 1, this is my anaphase 1, we have tetrads that are pulling apart from each other. So what may happen, so if that was my metaphase, during anaphase 1, you might get where this one separated just fine, the tetrad that was on the top, but maybe the one on the bottom, the whole tetrad started to move towards one side of the cell. So notice what this leads to is that you end up with, after we get telophase 1, you end up with this cell over here, which gets 3 chromosomes in it because we're looking at this one. And then this one over here is just going to have 1. So already after, this would be telophase 1, already after the very first meiosis, we've got this difference in chromosome numbers. We could have the same type of thing happen in anaphase 2. In anaphase 2, it's the sister chromatids that are supposed to separate. But again, if they don't separate from each other the right way, then too many move towards one side of the cell and too few move to the other side of the cell. So non-disjunction is going to give us really abnormal gametes. And if one of those abnormal gametes gets used for fertilization, then you have the wrong chromosome number in that final individual or in that zygote that is produced. So that's one type of chromosome abnormality. And that one, again, it's the wrong chromosome number. It's due to non-disjunction. Any of these individuals that have the wrong chromosome number, we call them aneuploids. So aneuploids, they could have one too many. They could also have too few chromosomes in them. And we can classify them further based on specifically what their abnormality is, if they have too many, too few, something along those lines. There are other chromosome abnormalities that are due to chromosome structure. And so this would be chromosome structure changes. And there's a lot of different categories that this falls into. We can have deletions of certain sections on the chromosomes. We can have what we call inversions, which would be where sections of the chromosome just get completely turned around so then they're in the opposite orientation that they should be. We can get pieces of the chromosome that actually get exchanged between non-homologous chromosomes. And that would not be something that's desirable, because recall that when crossing over takes place, crossing over is supposed to take place within the tetrad, and within the tetrad would be homologous chromosomes. And if you had a crossing over or an exchange of information between non-homologous chromosomes, we're not going to be exchanging the same information with each other. So these types of abnormalities, all of these down here, these tend to happen during DNA synthesis. So by DNA synthesis, we would be talking about S phase of interphase, when we're actually copying the DNA. So there could be some kind of problem there, and that problem is not caught during G2, so it's not fixed. Or if we're talking about something like this one down here, this could happen during meiosis. And this would be during really prophase 1, because we're talking about a problem with the crossing over process. And crossing over would take place during prophase 1.